So far, we've been talking about DNA in relation to human beings. And now we need to think about DNA in the other organisms that live on the planet. We need to do a domain comparison. As a review, there are 3 domains of living things that exist on this planet. There are bacteria, there are archaea, and there are eukarya. The bacteria and the archaea are called prokaryotes, and the eukarya are eukaryotes. And in terms of the DNA, there are a couple of important differences to remember. Prokaryotes do not have a nucleus. Their DNA is floating free within the cytoplasm of the cell. The DNA is also in a circular form. They have one chromosome, and it's in the form of a circle. Some bacteria also have some secondary DNA that's called plasmid DNA. Plasmids are simply small circles of DNA that a bacteria has that give it certain traits that are not necessary for survival, but can sometimes enhance survival. Plasmids often code for things like antibiotic resistance. Plasmids can also be transmitted from one bacterium to another, which is how bacteria can share traits like antibiotic resistance. Eukaryotes are very different. Eukaryotes have a nucleus, and that nucleus houses their DNA. Eukaryotes also have multiple chromosomes, and those chromosomes are linear. In other words, the 3 billion base pairs in a human being do not exist as a single chromosome. Instead, that one long molecule is broken up into smaller linear segments, and each segment contains a certain number of genes. Now, in human beings, these linear chromosomes form 23 pair, so a total of 46 chromosomes. 23 come from the mother, 23 come from the father. 22 of those pairs, or 44 chromosomes, are what we call autosomes, with auto meaning self and some meaning body. So these are chromosomes that contain genes that code for traits of the general body structure. Human beings also have a pair of sex chromosomes. If you are a male, you have one X sex chromosome and one Y sex chromosome. And if you're a female, you have 2 X chromosomes. Your sex chromosomes are unique. They are unlike your other chromosomes because they don't have to be a matched pair. Now in this slide, we've got a picture on the top that's depicting a generic bacterial cell. So you can see that this cell has an outer membrane, and it's got cytoplasm colored green on the inside. And then we've got this gold circular structure that represents the bacterial chromosome. Notice that it's circular. Notice that it is attached at one point to the wall, but the rest of the genetic material simply floats freely in the cytoplasm. This little extra piece of DNA over here is a plasmid. Remember, plasmids are not essential for life, and not all bacteria carry plasmids. Plasmids typically code for traits that give a bacterium a survival advantage. Down here on the lower part of the slide, what you're looking at is a highly magnified picture of a bacterial plasmid. So this is an actual plasmid inside a bacterial cell, and as you can see, it is circular. Now, in order to look at the chromosomes in a human being, we isolate them from the cell and we create what's called a karyotype, and that's what you're looking at in this slide. This is a karyotype that comes from a male, and I know that because in the lower corner here you can see the sex chromosomes. There's one X chromosome right here, and there's one Y chromosome right here. So this comes from a male. All the rest of the chromosomes are paired. One was inherited from mother, one was inherited from father, And if you look, you can see that there are 22 pairs. These are the autosomes. Now, each pair is identical one to the other, but if you go from pair to pair to pair, they're different. And you can see some of them are much shorter, and some of them are much longer. And each pair of chromosomes, compared to the other pairs, contains a unique set of genes. In other words, if you lined up chromosome 1, chromosome 2, chromosome 3, chromosome 4, all the way to chromosome 22, and the sex chromosomes, you would have the entire molecule of DNA from a human being. So again, when we talk about the eukaryotes, multi-celled organisms that have a nucleus inside of their cells, we're talking about chromosomes that are present within a nucleus. And one of the interesting things about chromosomes is that they're normally very dispersed within that nucleus. In other words, if we were to examine a eukaryotic cell underneath a microscope, we could see that there's genetic material in there if we stained for it, but we wouldn't be able to see individual chromosomes. It would just look like a nice uniform mass of material. There is one time, though, in a cell's life when chromosomes become visible underneath a light microscope, and that's during the time of cell division. The chromosomes undergo a process where they condense. They get shorter and they get fatter, and they become visible underneath the light microscope. So we've talked about what DNA is. We've talked about what it's made out of. We've talked about how it's structured from primary to secondary to tertiary structure. And I want to finish up this material by talking about what DNA is used for. Now, we've already talked about the central dogma. We've talked about how DNA is transcribed to make RNA, and RNA is translated to make protein. But that's only one of the uses of DNA. The other thing we have to remember is that DNA is used for a process called replication. Every time a cell divides and makes two new cells, the entire molecule of DNA has to be copied or replicated. This is a time when the cell performs a mechanism called DNA synthesis. The other use for DNA in the cell is the use we've already talked about, which is the production of protein. Again, DNA will be transcribed into a molecule called RNA. RNA will then be translated into protein. And it's protein, remember, that gives a cell its unique traits. So, you can think about replication or synthesis, as it's sometimes called, as a time when DNA is used to make more DNA in the cell. The DNA must be copied fully and completely every time a cell divides. That way, each new cell that's formed has a complete molecule of DNA in its nucleus. During DNA synthesis, the DNA molecule in the cell acts as a template to make more DNA. And I'll talk about that a little bit more in a minute. The other use for DNA in the cell is this process of transcription, where DNA is used to make RNA. In this process, the DNA again serves as a template, but it's not a template for making more DNA, it's a template for making RNA. And when we talk about transcription in the next lecture, we'll talk about the details of this important process called transcription. Now, one thing that's important to remember is that during DNA synthesis, the entire molecule of DNA has to be copied, and it has to be copied exactly. But during transcription, only a small portion of the DNA molecule gets transcribed into RNA. Only the regions of DNA that contain genes that produce a protein that the cell needs at that moment. So during transcription, only short sections of the DNA molecule are being transcribed to make RNA. In this slide, we can see a very basic image of cell division. Over on the left-hand side, you can see a generic animal cell, and it's got a black circle in the middle that represents the nucleus. The nucleus, again, is that region that harbors the DNA. During cell division, the first thing that has to happen is that the original cell, the parent cell, has to copy everything that's inside of it. All of the cytoplasm, all of the organelles, all of the dissolved molecules, and of course, all of the DNA. And that's what you see happening in this image. Then the cell undergoes a process called cytokinesis, where the cytoplasm divides, and that one parent cell becomes two new daughter cells. So, during DNA synthesis, when the nuclear material gets copied, the DNA molecule acts as a template. And the way it does this is by unwinding the DNA double helix, in other words, that belongs in the parent cell will separate into two strands. And that's what you're seeing in this slide. The light teal colored DNA of the parent cell has separated into two individual strands. Now, where you see this reddish pink strand of DNA forming, that's a brand new strand of DNA. And it's being made to be complementary to the original strand of DNA. In other words, when the double helix is separated and this strand of parental DNA is separated from this other strand of parental DNA, each of the base pairs is split. Everywhere on these exposed strands, then, there's room for the complementary base to come in and hydrogen bond. And that's what you're seeing happening here. Everywhere that there is an exposed T on the parental strand, an A deoxyribonucleotide can come in and bind. Everywhere there's an exposed G, a C deoxyribonucleotide can come in and bind. And slowly, bit by bit, as the double helix unwinds, new bases come in and are added on to this new strand. That process is happening on both of the original strands of DNA. And that's the process of DNA synthesis. This slide shows us that same process again in a different diagram. Here's the parental cell's DNA. It opens up into two white strands of DNA, and then complementary black strand bases come in and bind until we have two new molecules. We have this molecule of DNA and this molecule of DNA in each of the daughter cells. So in each daughter cell, the DNA molecule will contain one old strand, if you will, one parental strand, and one new strand that was made from that template. So DNA synthesis is a critical process in the life of a cell, anytime the cell divides. But DNA transcription is also a critical process. And if you stay tuned, that will be our next lecture topic.